Welcome to the second lecture. In this lecture, we will discuss the basic chemical components and principles that are relevant to the study of physiology. Remember that atoms and molecules make up cells, tissues, organs, organ systems, and organisms, all of which are the subjects of physiology. Therefore, learning physiology requires some familiarity with the basic concepts and terminology of chemistry. In this lecture, we will discuss the atomic and molecular structure, the nature of chemical bonds, and the nature of pH. We will also discuss the structure and functions of organic molecules, specifically the big organic molecules called macromolecules, including carbohydrates, lipids, proteins, and nucleic acids that make up the genetic material. Matter is anything that occupies space and has mass, including all living and non-living things in the universe. All matter is made up of these tiny particles called atoms. These particles are too small to be seen individually, even with the most powerful electron microscopes available today. Even though extremely small, atoms consist of three types of even smaller subatomic particles. The types of atoms vary in the numbers of these subatomic particles they contain. Protons and neutrons are particles of nearly identical mass, with protons carrying a positive charge and neutrons having no charge. Electrons have a smaller mass than protons and neutrons and are negative. An atom consists of two regions, a dense central nucleus made up of protons and neutrons surrounded by a three-dimensional electron cloud, where electrons move rapidly around the nucleus in orbitals. The magnitude of the charge of a proton exactly matches that of an electron, but it is opposite in sign, being positive. In all atoms, the number of protons in the nucleus is equal to the number of electrons moving around the nucleus, so their charges balance and the atoms are neutral. A pure substance composed of only one type of atom is called an element. A pure sample of element carbon contains only carbon atoms, even though the atoms might be arranged in the form of diamond or in the form of graphite. Of the 109 known elements, only 26 are normally found in the body. Four elements, hydrogen, carbon, oxygen, and nitrogen, comprise 96% of the body's mass. Because all matter is made up of atoms, atoms must somehow be held together to form matter. The forces holding atoms together are called chemical bonds. Not all chemical bonds are formed in the same way, but all involve the electrons of atoms. Whether one atom bonds with another depends on the number of and arrangement of its electrons. Electrons tend to move around in the nucleus in a specific pattern. The orbitals, or pathways traveled by electrons around the nucleus, are arranged in an orderly series of concentric layers, known as electron shells, which consecutively surround the nucleus. Each electron shell can hold a specific number of electrons. The first shell, closest to the nucleus, or the innermost, can contain a maximum of only 2 electrons, no matter what the element is. The second shell can hold a total of 8 more electrons. As the number of electrons increases with the increasing atomic number, which is the number of protons, more electrons occupy successive shells, each at a greater distance from the nucleus. For example, hydrogen here has only 1 electron, so that electron occupies the first shell. This shell can hold up to 2 electrons, and there is still a spot for another electron to fill. In contrast, the carbon here has 6 electrons. 2 of those electrons occupy the first shell, and the other 4 occupy the second shell. This means the second shell is short 4 electrons to completely fill it. Atoms tend to undergo processes that result in a filled outermost electron shell because a full outermost shell makes that atom stable. Thus, the electrons of the outer shell determine the bonding characteristics of an atom and its ability to interact with other atoms. Atoms that have a vacancy in their outermost shell tend to give up, accept, or share electrons with others, so that all participating atoms have filled their outer shells. For example, this carbon needs 4 more electrons to complete its outermost shell to become stable, whereas the hydrogen only needs 1 to complete its outermost shell. And so carbon recruits 4 hydrogen atoms that together can give that carbon 4 more electrons to completely fill the outermost shell. But this is a mutualistic relationship because the hydrogen atoms can also get the electrons from the carbon to completely fill its outermost shell to become stable. Each of these electron pairs forms what we call a covalent bond. Like carbon, other atoms can also share electrons to form chemical bonds. We call this type of bond covalent bond. A covalent bond is the sharing of a pair of outermost electrons by 2 atoms. The number of electron pairs that are shared between 2 atoms dictates the number of covalent bonds between them. For example, a pair of electrons shared between these hydrogen atoms results in a single bond, while 2 pairs of electrons shared between these oxygen atoms creates a double bond. 2 or more atoms held together by covalent bonds constitute a molecule. Atoms in a molecule attract shared electrons to varying degrees, depending on the element. The attraction of a particular atom for the electrons of a covalent bond is called electronegativity. The more electronegative an atom is, the more strongly it pulls the shared electrons toward itself. To help you recognize the relative electronegativity between some common elements, I've included this mnemonic here. It reads, flaming oxygen, nice clear bright, I suspect cooked ham. In this mnemonic, you see that fluorine is the most electronegative element, and hydrogen is the least electronegative element. In a covalent bond between 2 atoms of the same element, the electrons are shared equally between the 2 atoms because the 2 atoms have the same electronegativity, and so the tug of war between them is at a standoff. Such a bond is called a nonpolar covalent bond. For example, the single bond of H2, which is hydrogen element, is nonpolar, as is the double bond of O2. However, when one atom is bonded to a more electronegative atom, the electrons of the bond are not shared equally. This type of bond is called a polar covalent bond. Such bonds vary in their polarity, depending on the relative electronegativity of the 2 atoms. For example, the bonds between the oxygen and hydrogen atoms of a water molecule are very polar. An ionic bond is formed simply when a negatively charged atom is electrostatically attracted to a positively charged atom. We call this electrostatic interaction. There are two cases of ionic bond formation. In some cases, two atoms are so unequal in their attraction for the outermost electrons that the more electronegative atom strips an electron completely away from its partner. Specifically, the atom takes the electron from the other atom, now has an extra electron, and so it is negatively charged, or an anion. The atom that gives the electron becomes a positively charged species, and we call this a cation. A charged atom or molecule in general is called an ion. Once formed, the cation and the anion are bound together via electrostatic interaction to form ionic compounds. For example, this is what happens when an atom of sodium encounters an atom of chlorine. A sodium atom donates a single electron in its outermost shell and becomes a cation. The chlorine ion, or the chlorine atom, receives that electron and becomes an anion. Sodium chloride compound is formed when there is an electrostatic interaction between the sodium ion and the chloride ion. In other cases, the cations and anions have already been formed somewhere else through an electron transfer process. When these ions come into contact, they are attracted to each other through electrostatic interaction to form an ionic compound. The term ion also implies to the entire molecule that is electrically charged. For example, in the salt ammonium chloride, for instance, the anion is a single chloride ion, but the cation is the ammonium ion, which is positively charged. The whole ammonium ion has an electrical charge of 1 plus because it is 1 electron short. Weaker than ionic bonds are hydrogen bonds, which are the non-covalent attraction between a hydrogen and an electronegative atom, usually oxygen or nitrogen in living systems. For example, water contains an oxygen atom and 2 hydrogen atoms. Oxygen is one of the most electronegative of all of the elements, attracting shared electrons much more strongly than hydrogen does. Because electrons have a negative charge and are pulled toward oxygen in a water molecule, the oxygen atom has a partial negative charge indicated by the delta minus symbol, and each hydrogen ion or hydrogen atom has a partial positive charge indicated by the delta plus symbol. The slightly positive hydrogen of one molecule is attracted to the slightly negative oxygen of a nearby molecule. The two molecules are thus held together by a hydrogen bond, indicated by the dotted lines. 60% of your body is water because water is important for many processes. This sort of universality of water is due to the following properties, and let's examine them in detail. But first, for practice recognizing hydrogen bonds, see if you could determine which of the dashed lines between the molecules below are true hydrogen bonds. Water molecules stay close to each other as a result of hydrogen bonding. Although the arrangement of molecules in a sample of liquid water is constantly changing, at any given moment, many of the molecules are linked by multiple hydrogen bonds. These linkages make water more structured than most other liquids. Collectively, the hydrogen bonds hold the substance together, a phenomenon called cohesion. Cohesion is responsible for surface tension, which is the force that keeps a water surface from expanding or broken into. At the interface between water and air is the cohesiveness of water molecules forming hydrogen bonds with one another and to the water molecules underneath. This makes the water behave as though it is coated with an invisible film. You can observe the surface tension of water by slightly overfilling a drinking glass. The water will stand above the rim without spilling. In a more biological example, some animals can stand, walk, or run on water without breaking the surface. In contrast to cohesion, adhesion is the ability of water to form hydrogen interactions with other polar or charged substances. This property gives rise to the capillary action that helps draw blood passively up a capillary tube for blood sampling. This works by or because of the adhesion between blood, which is primarily water, and the walls of the capillary tube. This adhesion allows for water molecules in contact with the walls to climb up. In addition, because of cohesion, the whole water column climbs up with those water molecules up the capillary tube. The other special property of water is that it is able to act as a versatile solvent. Water solvation is the ability of water to surround another molecule and form chemical interactions with it. A liquid that is a completely homogeneous mixture of two or more substances is called a solution. The dissolving agent of a solution is the solvent, and the substance that is dissolved is the solute. An aqueous solution is one in which water is the solvent. Any substance that has an affinity for water is said to be hydrophilic or water-loving. This affinity is due to the hydrogen bonds between water and the charged or polar chemical groups of that substance. Therefore, but there are, of course, substances that do not have an affinity for water. Substances that are non-ionic or non-polar actually seem to repel water. These substances are said to be hydrophobic, which is water-fearing. An example from the kitchen is vegetable oil, which, as you know, does not mix with water-based substances like vinegar really well. Nonpolar molecules are parts of a molecule can aggregate together inside the chemical structure and away from the aqueous environment. This arrangement is more stable for them. The textbook calls this the hydrophobic interaction, but the reason why I would caution against this term interaction in this context is because interaction makes it sound like the nonpolar groups are actively binding to each other, but in reality they're not. A hydrophobic interaction is actually caused by the exclusion of nonpolar substances by water molecules. And so it's more of a reaction to being around water than an active binding and bonding activity. One way to remember this is that like dissolves like, meaning polar can dissolve in water, and likewise polar molecules, while nonpolar groups would not interact with polar groups and vice versa. Note that a compound does not need to be ionic to dissolve in water. Many compounds made up of non-ionic polar molecules, such as sugars, are also water soluble. Such compounds dissolve when water molecules surround each other of each of the solute molecules, forming hydrogen bonds with them. Even molecules as large as proteins can dissolve in water if they have ionic and polar regions on the surface. Many different kinds of polar compounds are dissolved in water of biological fluids like blood and the cytosol of all cells. The amount of solute dissolved in a specific amount of solution can vary. For example, a saltwater solution might contain 1 gram of salt in 100 milliliters of solution, or it can contain 10 grams of salt in 100 milliliters of solution. Both solutions are saltwater solutions, but they have different concentrations of solute. The concentration of a solution indicates the number of solute molecules in a volume of solution. Concentrations can be given in various units, and there are 3 ways to express the amount of solute in a solution. Concentrations given in terms of molarity give the number of moles of solute in exactly 1 liter of solution. Thus, a half molar or 0.5 m solution of NaCl or sodium chloride would contain 1/2 mole or about 29 grams of sodium chloride in each liter of solution. Molality, on the other hand, is the moles of solute per 1 kilogram of solvent. Both molarity and molality talk about the moles of solute in a solution. However, note that molality is different than molarity in that molality involves the weight of solvent instead of volume of solvent. Lastly, you can also express solute concentration based on a percentage. There are 3 ways to do this: mass percent solutions, volume percent solutions, or mass volume percent solutions. I'd like to point out that you get a lot more practice with calculating these types of concentration in lab. If you'd like to get more assistance on how to calculate this calculate solid concentration, please reach out to me, and we can work through some problems together. The bonds in water molecules joining hydrogen and oxygen atoms together are polar covalent bonds. These are pretty strong bonds, but sometimes a small proportion of those bonds do break. When that occurs, water molecules ionize to form hydroxide ions and hydrogen ions. We call this ionization of water. Because water ionization happens really rarely, the concentration of these ions in pure water is very low. In fact, in pure water, only 1 water molecule in every 554 million is dissociated. Actually, the concentration of each ion is therefore really low in pure water. It's about 10 to the negative 7 molars. And so by definition, a neutral solution is when the concentration of H plus is 10 to the negative 7 molars. In contrast, an acidic solution can release more H plus into a solution, causing that solution H plus concentration to be greater than 10 to the negative 7 molars. Basic solutions are those that can remove H plus ions from a solution, causing H plus concentration to be less than 10 to the negative 7 molars. We express the amount of H plus or the H plus concentration in water or aqueous solutions in terms of pH. By definition, pH is the negative log base 10 or natural log of the H plus concentration. And so for a neutral aqueous solution, H plus concentration is 10 to the negative 7, giving us a pH of 7. The internal pH of most living cells is close to 7. Even a slight change in pH can be harmful because the chemical processes of the cells are very sensitive to the concentration of H plus and OH minus ions. The pH of human blood is very close to 7.4 or slightly basic. A person cannot survive more than a few minutes if their blood pH drops to 7 or rises to 7.8. And a chemical system actually exists in the blood that can maintain a stable pH. The presence of substances called buffers allows biological fluids to maintain a relatively constant pH despite the addition of acids and bases. A buffer is a substance that minimizes changes in the concentration of H plus and OH minus in a solution. It does so by accepting H plus ions from the solution when they are in excess and donating H plus ions to the solution when they have been depleted. When there are several buffers that contribute to the pH stability in human blood and many other biological solutions. One of these is carbonic acid, formed when CO2 reacts with water in blood plasma. As mentioned earlier, carbonic acid dissociates to yield a bicarbonate ion and a hydrogen ion or H plus. If H plus concentration in the blood begins to fall, that is, if pH rises, the reaction proceeds to the right and more carbonic acid dissociates, replenishing hydrogen ions in the solution. But when H plus concentration in the blood begins to rise, i.e. when pH drops, the reaction proceeds to the left, producing more bicarbonate ions, the base, removing hydrogen ions from the solution to form carbonic acid. This exact buffer system in our blood helps to buffer H plus concentration produced by the ionization of metabolic acids. This way, we can homeostatically regulate blood pH so that it stays between 7.35 to 7.45. Living organisms, including humans, are made of chemicals that are based mostly on the element carbon. Remember that carbon has 4 electrons in its outermost shell and therefore can form 4 different chemical bonds with other atoms. This makes carbon a very chemically versatile element. Compounds that are composed of carbon and hydrogen atoms are called organic compounds. Proteins, DNA, carbohydrates, and other molecules are organic compounds that distinguish living matter from inanimate material. They are all composed of carbon atoms bonded to each other and to other atoms of other elements. The distinctive properties of an organic molecule depend not only on the arrangement of its hydrocarbon skeleton, but also the chemical groups attached to that skeleton. We can think of hydrocarbons, as the underlying network or framework for more complex molecules. A number of chemical groups can replace one or more of the hydrogens bonded to the carbon skeleton of the hydrocarbon. We call these chemical groups functional groups. These groups may participate in chemical reactions or may contribute to functions indirectly by their effects on the molecular shape. The number and arrangement of the groups help give each molecule its unique properties. Consider the differences between estradiol, which is a type of estrogen, and testosterone. These compounds are female and male sex hormones, respectively, in humans and other vertebrates. Both are steroids, which is a type of lipid, um, which we will discuss more in detail later. These sex hormones differ only by the chemical groups or functional groups attached to the rings. The distinctions in molecular architecture are shaded in blue here. Estradiol has a hydroxyl group as opposed to a carbonyl group as in the case of testosterone. In addition, testosterone has an extra methyl group. The different actions of these two molecules on many targets throughout the body help produce the contrasting anatomical and physiological features of male and female humans. Thus, even our sexuality has its biological basis in variations of molecular architecture. For this exercise, please circle and identify the functional groups present in each of these molecules. Indicate whether they are polar, nonpolar, or ionic. Given the rich complexity of life on Earth, we might expect organisms to have an enormous diversity of molecules. Remarkably, however, the critically important large molecules of all living things, from bacteria to elephants, fall into just four main categories: carbohydrates, lipids, proteins, and nucleic acids. On the molecular scale, members of three of these classes, carbohydrates, proteins, and nucleic acids, are huge and therefore called macromolecules. Moreover, each of these macromolecules is a polymer, which is a long molecule consisting of many similar or identical building blocks linked by covalent bonds. It's kind of like a train consisting of a chain of cars. The repeating units that serve as the building blocks of a polymer are smaller molecules called monomers. Although each class of polymer is made up of different types of monomers, the chemical mechanisms by which cells make and break down polymers are basically the same in all cases. In cells, these processes are facilitated by enzymes, which are specialized macromolecules that speed up chemical reactions. We will discuss in detail the structure and behavior of enzyme in the fourth lecture. Monomers are connected by a reaction in which two molecules are covalently bonded to each other with the loss of a water molecule. This is known as dehydration synthesis or dehydration reaction. When a bond forms between two monomers, each monomer contributes part of the water molecule that is released during the reaction. One monomer provides a hydroxyl group, while the other provides a hydrogen. This reaction is repeated as monomers are added to the chain one by one, making a polymer. Polymers are disassembled by into monomers by hydrolysis, a process that is essentially the reverse of the dehydration synthesis reaction. Hydrolysis means to break using water. So the bond between the monomers is broken by the addition of a water molecule, which with the hydrogen from the water attaching to one monomer and the hydroxyl group attaching to the adjacent monomer. An example of hydrolysis working within your body is the process of digestion. The bulk of the organic material in our food is in the form of polymers that are much too large for the cells to acquire. And so within the digestive tract, various enzymes attack the polymers. sort of speeding up the process of hydrolysis to break them down into monomers. And then the released monomers are then absorbed into the bloodstream and get distributed throughout the whole body. Carbohydrates include sugars and polymers of sugars. The simplest carbohydrates are monosaccharides or single sugar molecules. These are the monomers from which more complex carbohydrates are constructed. Disaccharides are double sugars consisting of two monosaccharides joined by a covalent bond. Carbohydrates also include macromolecules called polysaccharides, or polymers composed of many sugar building blocks. Monosaccharides generally have molecular formulas that are some multiple of the unit CH2O. Glucose, or C6H12O6, is the most common monosaccharide and is of central importance in biochemistry because it is the molecule of energy, and we'll learn why. A criterion for classifying sugars is the size of the carbon skeleton, which ranges from 3 to 7 carbons long. Glucose, fructose, and other sugars have 6 carbons, and so they're called hexoses. 3 carbon sugars are called trioses, and pentoses are 5 carbon sugars, and these are pretty common too. A disaccharide consists of 2 monosaccharides joined by a glycidic linkage, a covalent bond formed between 2 monomers by dehydration synthesis. For example, maltose is a disaccharide formed by the linking of 2 molecules of glucose. Also known as malt sugar, maltose is an ingredient used in brewing beer. And the most prevalent disaccharide, however, is sucrose, which is table sugar. The 2 monomers are glucose and fructose. Lactose, on the other hand, is the sugar present in milk, and it's another disaccharide, but in this case, it is a glucose molecule joined to a galactose molecule. You don't have to know the structures of these sugars besides glucose. Polysaccharides are macromolecules or polymers with a few hundred to a few thousand monosaccharides joined by glycosidic linkages. Some polysaccharides serve as storage material, hydrolyzed as needed to provide sugar for cells. Other polysaccharides serve as building material for structures that protect the cell or the whole organism. Both plants and animals store sugars for later use in the form of storage polysaccharides. Plants store starch, a polymer of alpha glucose monomers. Most of the alpha glucose monomers in starch are joined by the 1 to 4 glycosidic linkages. Number 1 carbon to number 4 carbon, that is. Just like the glucose units in maltose. The sugar can later be withdrawn from this carbohydrate bank by hydrolysis, which breaks the bonds between the glucose monomers. Most animals, including humans, also have enzymes that can recognize 1,4 glycosidic linkages between alpha glucose monomers in starch and can hydrolyze them, thereby making glucose available for nutrients as nutrients for cells. In addition, humans store excess glucose in the form of glycogen, a polymer of glucose that is like starch but more extensively branched. Humans and other animals store glucose mainly in the liver and muscle cells. Okay, in terms of structural polysaccharides, organisms build strong materials from structural polysaccharides. For example, the polysaccharides used by plants to make cellulose is a major component of the tough walls that protect plant cells. Like starch, cellulose is a polymer of glucose, but the glycosidic linkages in these two polymers differ. The difference is based on the fact that monomers of cellulose is of beta glucose instead of an alpha glucose as in the case of starch. What is the difference between the two kinds of glucose? When glucose forms a ring, the hydroxyl group attached to the number 1 carbon is positioned either below or above the plane of the ring. If it's below, then it's an alpha glucose. If it's above, then it's a beta glucose. Enzymes that digest starch by hydrolyzing its alpha linkages are enabled to hydrolyze the beta linkages of cellulose because of the distinctly different shapes of the two molecules. In fact, only a few organisms possess enzymes that can digest cellulose. Animals, including humans, do not. The cellulose in our food passes right through the digestive tract and is eliminated with the feces. Along the way, cellulose abrades the wall of the digestive tract and stimulates the lining to secrete mucus, which aids in the smooth passage of food throughout the tract. Lipids are the one class of large biological molecules that does not include true polymers. They are generally not big enough to be considered macromolecules. Compounds called lipids are grouped together because they share one important trait, and that is they mix poorly, if at all, in water. The hydrophobic behavior of lipids is based on their molecular structure. Although they may have some polar bonds associated with oxygen, lipids consist mostly of hydrocarbon regions. Lipids are varied in form and function, but we will focus on the most biologically important ones, including triglycerides, phospholipids, and steroids. A triglyceride is constructed from two kinds of smaller molecules, glycerol and fatty acids. Glycerol is an alcohol. Each of its 3 carbons has a hydroxyl group. A fatty acid has a long carbon skeleton and a carboxyl group at the end of the chain. The relatively nonpolar CH bonds in the hydrocarbons, or the fatty acids actually, are the reason why triglycerides are actually hydrophobic and separate from water. In making a triglyceride, 3 fatty acids are each joined to glycerol by dehydration synthesis to form 3 linkages between the hydroxyl groups of the glycerol and the carboxyl groups of the fatty acids. The resulting triglyceride thus consists of 3 fatty acids linked to 1 glycerol molecule. There are 2 types of fatty acids, unsaturated fats and saturated fats. If there are no double bonds between the carbon atoms comprising a chain, then as many hydrogen atoms as possible are bonded to the carbon skeleton. Such a structure is said to be saturated with hydrogen atoms, and the resulting fatty acid is therefore called saturated fatty acid. The unsaturated fatty acid has 1 or more double bonds with 1 fewer hydrogen atom on each double bonded carbon. And so nearly all double bonds in naturally occurring fatty acids are in cis configuration, which causes a kink in the hydrocarbon chain wherever they occur. A cis double bond is when both chemical groups attached to the double bond are on the same side of it. A fat made from saturated fatty acids, it's called saturated fat. Most animals are saturated fat. The hydrocarbon chains of their fatty acids, or the tails of the fat molecules, lack double bonds. And so their flexibility allows for the fat molecules to pack together tightly. Saturated animal fats such as lard and butter are solid at room temperature. In contrast, the fats of, say, plants and fishes are generally unsaturated. meaning they are built of one or more types of unsaturated fatty acids. Usually liquid at room temperature, plant and fish fats are referred to as oils. So olive oil and cod liver oil are examples. The kinds of the kinks where the cis bond, the double bonds are located, prevent the molecules from packing closely together enough to solidify at room temp. Cells do not exist without another type of lipid called phospholipids. Phospholipids are essentially for cells to uh, make their cell membranes. Their structure provides a classic example of how form-fits function at the molecular level. As shown in this figure, the top figure, a phospholipid is similar to a fat molecule, but has only two fatty acids attached to the glycerol rather than three. The third hydroxyl group of the glycerol is actually joined to a phosphate group, which has a negative charge in the cell. Additional small variable molecules, which are usually charged or polar, are linked to the phosphate group to form a variety of phospholipids. Most abundant is phosphatidylcholine, which is in which the variable group is choline. The two ends of phospholipids show different behavior toward water. The hydrocarbon tails are hydrophobic and are excluded from water. However, the phosphate group and its attachments form a hydrophilic head that has an affinity for water. When phospholipids are added to water, they self-assemble into double-layered structures called bilayers, shielding their hydrophobic portions from water. Steroids are lipids characterized by a carbon skeleton consisting of four fused rings. Different steroids such as cholesterol and sex hormones are distinguished by the particular functional groups attached to the ensemble of rings. And cholesterol is a crucial molecule in animals. It is a common component of animal cell membranes and is also the precursor from which other steroids such as sex hormones, mineralocorticoids, glucocorticoids are synthesized. In humans, cholesterol is synthesized in the liver and or obtained from the diet. A high level of cholesterol in the blood may contribute to atherosclerosis. And in fact, both saturated fats and trans fats exert their negative impact on health by affecting cholesterol levels. Nearly every dynamic function of the living system depends on proteins. Some proteins speed up chemical reactions, while others play a role in defense, as in antibodies, cellular communication, as in hormones, and movement, as in muscle tissue. Proteins are polymers constructed from the same set of 20 amino acids monomers. The polymers of amino acids are called polypeptides. A protein is a biologically functional molecule that consists of one or more polypeptides, each folded and coiled into a specific three-dimensional structure. All amino acids share a common structure. At the center of the amino acid is a carbon atom called the alpha carbon. Its four different partners are the amino group, carboxyl group, a hydrogen atom, and a variable group symbolized by R. The R group, also called the side chain, differs with each amino acid. The physical and chemical properties of the R group determine the unique characteristics of a particular amino acid, thus affecting its functional role in a polypeptide. In this figure, all the way on the right, the amino acids are grouped according to the properties of their side chains. One group consists of amino acids with nonpolar side chains, which are hydrophobic. Another group consists of amino acids with polar side chains, which are hydrophilic. Acidic amino acids are those with side chains that are generally negative in charge owing to the presence of a carboxyl group, which is usually dissociated um, or ionized at cellular pH. Basic amino acids have amino groups in their side chains that are generally positively charged. Because they are charged, acidic and basic side chains are also hydrophilic. In addition, if you see hydrocarbon rings, then chances are it is a nonpolar and hydrophobic. And this is because if the chemical structure is symmetrical, as in a ring, then the electrons are equally distributed across the ring, and there is no polarity among the atoms. You must learn how to recognize basic, acidic, polar, and nonpolar side chains. Now that we have examined amino acids, let's see how they are linked to form polymers. When two amino acids are positioned so that the carboxyl group of one is adjacent to the amino group of the other, they can become joined by a dehydration reaction with the removal of a water molecule. The resulting covalent bond is called a peptide bond. Repeated over and over, this process will produce a polypeptide, which is a polymer of many amino acids linked together by peptide bonds. In spite of their great diversity, all proteins share three levels of structure known as primary, secondary, and tertiary structure. A fourth level, or the quaternary structure, arises when a protein consists of two or more polypeptide chains. The primary structure of a protein is simply a linked series of amino acids with a unique sequence. Most proteins have segments of their polypeptide chains repeatedly coiled and folded in patterns that contribute to the protein's overall shape. These coils and folds collectively referred to as the secondary structure, um, which are which is the result of hydrogen bonds between the repeating constituents of the polypeptide backbone, not the amino acid side chains. Within the backbone, the oxygen atoms have a partial negative charge, and the hydrogen atoms attached to the nitrogens have partial positive charge. Therefore, hydrogen bonds can form between these atoms. Individually, these hydrogen bonds are weak, but because they are repeated many times over a relatively long region of the polypeptide, they can support a particular shape for that part of the protein. One such secondary structure is the alpha helix, a delicate coil held together by hydrogen bonds. The other main type of secondary structure is the beta pleated sheet, also shown here. While secondary structure involves interactions between the backbone constituents, tertiary structure is the overall shape of a polypeptide resulting from interactions between the side chains or R groups of the various amino acids. One type of interaction that contributes to tertiary structure is hydrophobic interaction. A polypeptide folds into functional shape. When that happens, amino acids with the hydrophobic or nonpolar side chains usually end up at clusters at the core of the protein, out of contact with water. Once nonpolar amino acid side chains are close together, the 3D structure is stabilized by many other weak non-covalent bonds, such as ionic bonds, or hydrogen interactions, and sometimes a few covalent bonds, namely the disulfide bonds. When a cell synthesizes a polypeptide, the chain generally folds spontaneously, assuming the functional structure for that protein. The folding is driven and reinforced by the formation of a variety of bonds between parts of the chain, which in turn depends on the sequence of amino acids. Many proteins are roughly spherical, um, which make for globular proteins, while others are shaped like long fibers, and so they're fibrous proteins. The basic unit of fibrous protein is a polypeptide that is composed of repeating elements of secondary structure arranged in a long strand. Multiple strands twisted around each other create a fibrous molecule. This setup allows for fibrous proteins to resist mechanical stress, such as tension and compression. Examples include collagen, elastin, and keratin. In contrast, many globular proteins have a quaternary structure because they're composed of multiple subunits of tertiary structure. An example includes hemoglobin. In addition, some globular proteins are made of only one polypeptide that is folded into a tertiary structure. An example is myoglobin, which is the sister version of hemoglobin in your muscles. Globular proteins are also pretty diverse in shape compared to fibrous proteins. There's not much diversity with fibrous proteins because you can make it thicker or longer, but that's about it. In contrast, with globular protein, you can mold it into different shapes depending on the R group interactions. It is almost like molding Play-Doh into different shapes. You'll learn, you see later that the diversity in shape allows globular proteins to recognize and interact with a variety of proteins, especially in enzymes. Remember that protein structures are held together via weak chemical interactions, which are susceptible to changes in the physical and chemical conditions of the protein's environment. If the pH, salt concentration, temperature, or other aspects of its environment are changed, the weak chemical bonds and interactions within a protein may be destroyed, causing the protein to unravel and lose the native shape. A process called denaturation. Because it is misshapen, the denatured protein is biologically inactive. Most proteins become denatured because, or if they are transferred from one aqueous environment to a nonpolar solvent, such as ether or chloroform. The polypeptide chain refolds so that its hydrophobic regions face outward toward the solvent. This would change the shape of protein and therefore its function. Other denaturation agents include chemicals that disrupt the hydrogen bonds, ionic bonds, and disulfide bonds that maintain a protein's native shape. For example, altering the pH can change the concentration of H plus, and having more H plus can, say, neutralize basic R groups, which would destroy the existing electrostatic interactions that contribute to protein folding. Denaturation can also result from excessive heat, which makes the polypeptide chain become agitated, and they move enough that they overpower the weak interactions that stabilize their structure. This is because high temperature increases the movement, and what we call kinetic energy, which can increase the movement of atoms and disturb the weak interactions. For example, the white of an egg becomes opaque during cooking because the denatured proteins are insoluble and solidify. This is this also explains why excessively high fevers can be fatal. Proteins in the blood can denature at a very high temperature. If the primary structure of polypeptides determines the protein's shape, the question is, what determines the primary structure of polypeptides in the first place. The amino acid sequence of a protein is actually encoded in your genetic material, which is made of DNA or deoxyribonucleic acid. DNA belongs to the class of compounds called nucleic acids, which are polymers made of monomers called nucleotides. A DNA strand is composed of a series of DNA nucleotides. A pair of DNA strands makes up a DNA molecule. If you think of DNA as a ladder, then the sugar and phosphate components of the nucleotides form, the struts, the vertical struts of the ladder, while the pairs of the nucleotide bases interconnected by, in this case, hydrogen bonds, form the horizontal rungs. When a cell is not dividing, the DNA and its associated proteins called histones are in the form of an unwound mass of DNA material called chromatin. It's kind of like a bowl of cereal, a bowl of spaghetti. Once the cell begins to divide, the chromatin rearranges itself in a more precise and identifiable elongated structure. And multiple structures like this are called chromosomes. The chromosomes are shown in different colors here in this figure. Each chromosome contains a single long molecule of DNA and associated with it proteins histones. Each of our cells contains 46 chromosomes, 23 from your mom and 23 from your dad. The other type of nucleic acid is called RNA, or ribonucleic acid, which is also made of nucleotide subunits. RNA carries the instructions for protein synthesis encoded in DNA. A segment of a DNA strand that encodes for a specific protein is called a gene. The process of reading the instructions built in your DNA to make a protein is called gene transcription, or gene expression rather. Because DNA in the form of chromatin is a little bit too chunky to leave the nucleus to give instructions to the ribosomes, whenever a cell needs to make a protein, a copy of DNA has to be made. It has to be converted to RNA. This conversion is called transcription. Because RNA is only a short sequence of nucleotides carrying instructions for creating a specific protein, it is small enough to be transported out of the nucleus and into the cytoplasm. There, it will be read by the ribosomes to make proteins. The process of making proteins from RNA is called translation. Imagine this. If your DNA is like a cookbook and it contains genes that are like recipes. Say if you cannot take the book home from the cookbook home from the library, so in order to cook the dish, you need to make a photocopy of the recipe. This photocopy is like RNA, and the process of making a photocopy is like transcription. The process of following the recipe in your kitchen is comparable to translation, and the dish itself is like a protein. A nucleotide in general is composed of three parts: a pentose, or a 5-carbon sugar, a nitrogen-containing base, or a nitrogenous base, and 1 to 3 phosphate groups. The beginning monomer used to build a polynucleotide has 3 phosphate groups, but 2 are lost during the polymerization process. To understand the structure of a single nucleotide, let's first consider the nitrogenous bases. Each nitrogenous base has 1 or 2 rings that include nitrogen atoms. There are two families of nitrogenous bases, pyrimidines and purines. A pyrimidine has one six-membered ring of carbon and nitrogen atoms. You don't have to memorize these structures, but know that the members of the pyrimidine family are cytosine, abbreviated as C, thymine or T, and uracil or U. Purines are larger with a six-membered ring fused with a five-membered ring. Again, you don't have to know the structure of this, but the purines are adenine and guanine, abbreviated as A and G, respectively. The specific pyrimidines and purines differ in the chemical or functional groups attached to the rings. Note that A, C, and G are found in both DNA and RNA, but thymine is found only in DNA and uracil is only found in RNA. Well, now let's add the sugar to which the nitrogenous base is attached. In DNA, the sugar is deoxyribose. In RNA, it is ribose. The only difference between these two sugars is that deoxyribose lacks an oxygen atom on the second carbon in the ring, hence the name deoxy and deoxyribose. To complete the construction of a nucleotide, we attach 1 to 3 phosphate groups to the 5' carbon of the sugar. The carbon numbers in the sugar include the prime symbol. Now we can have a complete nucleotide, which includes a nitrogenous base, a sugar, and a phosphate. The linkage of nucleotides into polynucleotide in other macromolecules involves a dehydration synthesis reaction. In the polynucleotide, adjacent nucleotides are joined by a phosphodiester linkage, which consists of a phosphate group that links with the two sugars of the two nucleotides. DNA molecules have two polynucleotides or strands that wind around an imaginary axis, forming a double helix. The sugar phosphate backbones are on the outside of the helix, and the nitrogenous bases are paired in the interior of the helix. The two strands are held together by hydrogen bonds between the paired bases. In base pairing, only certain bases in the double helix are compatible with each other. A always pairs with T, and G always pairs with C. Reading the sequence of bases along one strand of the double helix would tell us the sequence of bases along the other strand. For example, if a stretch of one strand has the base sequence GCAC, then the base pairing rules tell us that the same stretch on the other strand must have the sequence CGTG. The two strands of the double helix are thus complementary. Each is the predictable counterpart of the other. In this feature of DNA that makes it is this feature of DNA that makes it possible to generate two identical copies of each DNA molecule in a cell that is preparing to divide. When the cell divides, the copies are distributed to the daughter cells, making them genetically identical to the parent cell. Thus, the structure of DNA accounts for its function in transmitting genetic information whenever a cell procreates or a cell reproduces. RNA molecules, by contrast, exist as single strands. Complementary base pairing can occur, however, between regions of two RNA molecules or even between two stretches of nucleotides in the same RNA molecules. In fact, base pairing within an RNA molecule allows it to take on the particular 3D shape necessary for its function. Consider, for example, this particular type of RNA called transfer RNA or tRNA, which brings amino acids from the cytoplasm to the ribosome during the synthesis of a polypeptide. tRNA's functional, its functional shape results from the base pairing between the nucleotides of complementary stretches within the molecule. It is this 3D shape that allows the tRNA to be recognized by and bind to ribosomes, carrying amino acids to the site of protein synthesis. Note that in RNA, A always pairs with U, and T is actually not present in RNA. Another difference between RNA and DNA is that DNA almost always exists as a double helix, whereas RNA molecules are more variable in shape. Note that the sequence of bases along a DNA or RNA for that matter is unique for each gene and provides very specific information to the cell. Because genes are hundreds to thousands of nucleotides long, the number of possible base sequences is effectively limitless. The information carried by the gene is encoded in its specific sequence of the 4 DNA bases. The linear order of bases in a gene specifies the amino acid sequence, the primary structure of a protein, which in turn specifies that particular protein's 3D structure, thereby enabling its function in the cell. Besides serving as the building blocks of your genetic material, nucleotides can be converted into several derivatives or structures that derive from the nucleotides. The two that we care about are ATP, or adenosine triphosphate, and cyclic adenosine monophosphate, or CAMP. ATP contains the sugar ribose with the nitrogenous base adenine and a chain of 3 phosphate groups, the triphosphate group bonded to it. The bonds between the phosphate groups of ATP can be broken by hydrolysis. When the terminal phosphate bond is broken by addition of a water molecule, a molecule of inorganic phosphate leaves the ATP, which becomes adenosine diphosphate, or ADP. This hydrolysis at the terminal phosphate bond releases energy, which can then be harnessed to do work in the body. A derivative of ATP is CAMP. CAMP is very commonly used in intracellular signaling programs. It acts as a signal molecule that is used within the cell. Notice that cyclic AMP is removing 2 out of 3 phosphate groups and form a phosphodiester linkage between the 5' and 3' carbons. This phosphodiester linkage creates a new ring in the structure, in addition to the ribose and the nitrogenous bases. This ring accounts for the cyclic part of AMP, or cyclic AMP. CAMP can then be converted into AMP by an enzyme called phosphodiesterase. This is a really important enzyme, so make sure you understand how this works, or actually what it does, not the enzymatic mechanism, not yet. For signaling pathways that use CAMP, the phosphodiesterase effectively gets rid of CAMP and turns it off, so that the signaling program is no longer active. Okay, so that was it. Sorry about the first version of this, which was really long-winded, and I got pretty much really carried away with all of the biochemistry. But like always, please use these objectives to guide your preparation for the quiz, and also remember to submit the anonymous questions on Google Docs. See you in class.